Okay, so we've generated an overview, and now we have a bunch of things we can do. It's actually going to tell us some of the possibilities: assign probable enzymes, rescore pathways, assign modified proteins, create complexes, and run the consistency checker. What we're going to do right now is save this. So up to Organism, save KB, and KB is knowledge base here, which is another way of saying database. So let's save. Okay, our database is saved. We have a rudimentary model organism database now. So let's click here and exit. Now I clicked home there just to refresh this overall organism menu. And now you can see we've added our Coxiella burnettii Dugway 7E912 database. Our summary tells us we have 140 pathways right now. We have a little over 2,000 genes. A lot of citations, those are pulled in from BioCyc, so let's click on that. Here's our overview: 737 enzymatic reactions, 2,140 polypeptides, etc., etc. Let's go to Overview and show the cellular overview. And there we go. There is the state of our cellular overview right now. Now this is actually going to change as we make more modifications to the database, because right now we've predicted reactions and we've predicted pathways, but we haven't done anything else. So let's go back and refine. Tools, again, pathologic. Note that our database is still selected, and it's going to stay selected as long as we keep the application open until we tell it to pick a different database. Refine, assign probable enzymes. Okay, so what this does is it pulls up a list of things that it thinks are enzymes, but it couldn't be exactly sure. And often what will happen here is we have, for example, something that says, hey, this is an ACE. That sounds like an enzyme. Is that an enzyme? Can you assign it? And you can see here, well, these are just called acetyltransferase, some family. We probably can't assign those. But this is 2-polyprenyl 6-methoxyphenyl 4-hydroxylase. We might be able to assign that. So what you do is you go through these things one at a time, left click on it, and you get this probable enzyme status dialog. Now, what it's going to do is it's going to say, sometimes it'll just be saying, I don't know, but this seems like an enzyme. Sometimes it'll do this. Hey, I think it might be this. Is it this? Is it this? And we're going to say, yeah, it's that. So let's assign that. And it shows you a little bit of the protein frame. You'll see a little bit more of this later when we go to editing. And you can say, okay, yeah, that looks good. Okay, that looks good. Boom. Now it's assigned. And so we can continue running through and doing the same thing. Assigning. Yes. Okay. And we have, in this case, 143 of these that we can assign. So let's look at this thing, this acetyltransferase GNAT family. We can say, there's no thing I can assign it to. Notice there's an assignment comment here, so when I make the assignment I can write a little note about it for later to remind me, why did I do this? So I can say, in this case, this is not a specific enzyme, I can't assign it. So, okay. And boom, that blanketed all these because they were the same name. Acetyltransferase family protein, kind of the same thing. You're going to hit a lot of these non-specific things, and again, that's purely a function of the annotation. So if you did the annotation, it's your fault, and otherwise it's just you're relying on what the annotators put in. You know, so you have a number of options. Let's pull that up. In addition to just picking one of the reactions that it's guessing at for you and saying sure, you could also freely assign it to a reaction that's already in the database, or even pull something in from BioCyc. But I'm just going to click yes here. assign, and when it actually brings up this window, you can put in things like citations. So if you're sitting here with a stack of literature or with PubMed open in your browser and you're looking through on a protein by protein basis as you do this and finding literature that supports this, your annotations, your links, you can include that right here. And there'll be, again, a little more on that in the second round of this webinar series where we talk about editing. Okay? So basically, you just run down the list, do this kind of thing, and of course I'm skipping around like mad here, but you can do it whichever way you like. And I am going to exit. Actually, I'm going to save the KB first, then I'm going to exit. I'm not going to run through all these right now, but you can do this. You can always come back and do this later for the things that haven't been assigned. So let's exit now. And it's going to save again on the exit. There we go. Okay. I made some changes, so I may want to rescore the pathways. So refine, rescore pathways. Let's do that. So what it's doing now is I may have added in some new enzyme activities, which I did, and I haven't taken any out, but this may affect whether or not our software believes that certain pathways are more or less likely to actually exist in your organism. I made very few changes, so probably there's very little difference, but I'm going to go ahead and do it by way of example now anyway. Okay, so that's done. Additional refinement. So we've made the database, we've assigned probable enzymes, some of them at any rate. Refine. What else do we have? We can assign modified proteins. I'm not going to do that now, but that'll let you put in things like a phosphorylated state of a certain protein, create protein complexes. This is an important thing to do if you have a lot of data about protein complexes in your organism of interest, because we don't predict very many protein complexes at all. If you were noticing when we went through the summary screen, for Coxiella here we have 4 predicted. And that's basically because there's usually not a lot of information with which to predict protein complexes looking just at the genome annotation file. The annotation typically doesn't support it. We could rerun the name matcher. I'm not going to do that, but that would look again at all the enzyme names, or the protein names rather, and see if it thinks any of them are enzymes. We can rescore pathways, which we just did. We can predict transcription units. We're going to do that now. Our next step after this is predicting transporters, or actually identifying transporters. And first we want to do transcription units. So let's start with that. Click on that. Select for TU prediction. We're going to select all because we want to predict transcription units both in the chromosome and in the plasmid. And we click predict, and it starts. Okay, and now we've predicted transcription units. You may notice this note at the bottom. It is recommended that you rerun the predictor after rescoring pathways, or if the knowledge base has undergone changes in pathways and protein assignments. And that's because part of what's involved in predicting transcription units is looking at whether multiple genes that are adjacent to each other are also involved in the same pathway. Now, we'll refine again with the transport identification parser. And what this is doing is it's just looking through all of the protein names for candidate transporters and trying to figure out if you may have transporters in your organism so it can start attaching transport reactions to them. And it will just keep track for you at the bottom of the page here how many candidates it's looking through out of the total candidates it has. So it has 127 candidate transporters to look through right now, and we're at about 45. So it's a pretty fast process.